Are you guys as excited to talk about the heart as I am? No? Oh, come on. It's such a cool tissue. It's not working then? All right. So this isn't working. Okay. I guess I got to stand behind. There we go. Okay. So let's talk about the heart from the standpoint of tissues. What are, good, it's not up there. What are the 4 tissues in the body? Muscle, nervous, and then, yeah, all right, good. All right, so epithelial tissue, connective tissue, muscle tissue, and then nervous tissue. So this is going to be one of our muscle tissues. Right, so some of the things that we discussed about the cardiac muscle is that it's only located in one place. We're going to go and review and talk about, this slide should look a little bit familiar to you, right, because we talked about it briefly in lab. So I want to go over it again because it's important that you understand the muscle cell anatomy, because we're going to get into the actual cellular physiology of how these muscle cells contract, what stimulates that, you know, and you learned some of that in 210, but we're going to move into it in a lot more detail here in 211. So keep in mind, when we talk about the myocardium, where's that located? Don't say the heart because we already know that, but where in the heart? Specifically. Right, the middle layer, nailed it. Yep, it's the middle layer, it's the heart wall. Okay, so that's where we're going to see it. It's the only place in the body. Is it voluntary or involuntary? Involuntary, good, good. All right, so review about the muscle cells for the cardiac cells. They are branch cells. They're not going to be long or elongated as the skeletal muscle is, and they'll have 1 to 2 nuclei. And so they'll also be striated. So that means if there's striation characteristics to this muscle cell, then that means, right, we know the orientation of the actin myosin. It's going to be in those sarcomeres. So we're going to have sarcomeres that we're going to be dealing with here. Right, so when we name the plasma membrane, we call that the sarcolemma. That's all review. Y'all should remember that. The plasma membrane is the sarcolemma, and then you'll have these holes throughout the sarcolemma, and those are the openings to the T tubules, and that's where our action potential is going to travel along the sarcolemma, and then like water going down the drain in the bathtub, the action potential is going to go down into those openings for the T tubules and start triggering the opening to calcium-sensitive gates, which will cause the release of calcium from the sarcoplasmic reticulum, which is this fella right here. All right, so your sarcoplasmic reticulum is that tubular network that is going to be associated with these myocardial cells. And what's in the sarcoplasmic reticulum that is so important that the cell needs? Calcium. Yep. Okay, so keep in mind, the sarcoplasmic reticulum is separated from the sarcoplasm. The sarcoplasm is the name of the cytoplasm for a cardiac muscle cell. You should all know this. All right, so what's in the sarcoplasm? The actin and myosin filaments, the contractile filaments. Right, so, and the sarcoplasmic reticulum is separate, and that's where the calcium is, right, going to be located in between muscle contractions, because we need calcium to initiate a muscle. Well, we don't need, well, yeah, we do need it. We need calcium to initiate a muscle contraction because that's going to remove, remember, the troponin, tropomyosin, regulatory proteins that sit on the actin molecule. All right, so keep in mind when we're looking at this tissue, it has sarcomeres, therefore it has a striated appearance. And this is what I was telling you about. We'll talk about it later on in this chapter about the Frank-Starling law. Right? But when we talk about optimal length, this is that Goldilocks effect. Right? So when those contractile proteins are interacting with one another, if you have too much overlap, you're not going to get a very good contraction. If you have too little overlap, you're not going to get a very good contraction right in the middle. Right? We want it to be in the middle where we have optimal or maximum overlap, right, where it's not too much. Okay, so I call it the Goldilocks effect here. And so that allows then when those muscle cells contract, it allows those muscle cells to contract with greater force, because obviously the more overlap or connections that you can form between actin and myosin, you can get a stronger muscle contraction. You get a stronger muscle contraction, we can generate more force. We can pump more blood. Because that's the job of the heart, pumping blood. in one direction. Okay. So some of the things that we're going to see, and we, again, very important, the intercalated discs, right, which are made up of desmosomes and gap junctions. The desmosomes are going to help achieve adhesion, effective, optimal adhesion between the cells. So when one cell starts to contract, it'll help the other cell to contract. And so this offers those tissues much more stability so it can increase the effectiveness of those contractions. And then the gap junctions, you all remember, right? Those are essentially holes, right, in between cells. So ions, and we know some of these ions, calcium, potassium, sodium, so they can travel both outside the cell, but the gap junctions allow them to flow through the cell with less resistance, so we can get a better, quicker contraction if need be. And so that brings us to this phenomenon here called the functional syncytium, and this allows each chamber of the heart to essentially contract at the same time. Right, so the top part of the right ventricle and the bottom part of the right ventricle will essentially contract at the same time due to these intercalated discs, which is good. You saw in that video there, that heart contraction video, right? We saw the atria contract at the same time and the ventricles to contract at the same time. You need to have that coordination there, because if it's all off, then we'll start to have some pathologies that can occur, affect blood flow, can start to get clotting, can get damage to cells. It's a whole slew of issues. So we want to make sure that we're not able to run into these problems. So these intercalated discs with the desmosomes and the gap junctions help with that. So that's what you're seeing here in this little box. Here you can see those gap junctions, usually 6 proteins that are in a concentric circle here that allow the ions to flow from one cell to the other. And then we have our desmosomes here that help to anchor the cells to one another so they can effectively contract more efficiently. So this slide here is showing us, again, I'm not going to go and review the whole sarcomere anatomy with H zones and Z discs and all that. If you want to review that, it's in chapter 10. All right, but same kind of configuration, because we're dealing with sarcomeres, but we'll have, again, keep in mind, the sarcomeres were the contractile filaments. Those reside in the sarcoplasm. If you didn't know that before, make sure you know it now. And they're separate, all right, from the sarcoplasmic reticulum. That's that tubular network that you see all over the place there. That's where the calcium is going to reside when it's not in the sarcoplasm, causing the contractile filaments to have those cross-bridging interactions. All right, so nice review there. Now, how do we power this thing? Well, we're going to power your heart much like we do most of our skeletal muscle. I mean, like we power a lot of our skeletal muscle with ATP. All right, and so these heart cells have a small amount of ATP on hand in the sarcoplasm, but that gets used up relatively quickly. Right, so we have to be able to generate ATP, and I'll talk about that in a second here. But when we're looking at this tissue, notice you're going to have lots of mitochondria, right? Quite extensive blood supply to those cells, right? So the mitochondria are for what? ATP respiration, right? Specifically aerobic cellular respiration. Aerobic cellular respiration. What does aerobic mean? With oxygen. Yeah, so we need that oxygen. So we learned in the previous chapter that oxygen is going to attach itself onto hemoglobin, which is the oxygen, or excuse me, the respiratory gas carrying protein in our blood. Guess what? Your cardiac cells have their own version, and you were introduced to this before, called myoglobin. Myo, think muscle. And so it has its own oxygen, all right, binding molecule or protein in the sarcoplasm. And then also, we can help out that cell, because if you have a nice, extensive blood supply that is going to be bringing blood to these tissues, that means you're going to have a lot of hemoglobin on the red blood cells, so you'll have a pretty decent amount of oxygen. So that's important that you have that. I know I talked about this in lab, that creatine kinase, right? Kinase is an enzyme that likes to put phosphate onto things. So we call that process phosphorylation. And so that's what this, right, molecule does. It will put phosphate, it'll take it off of a molecule called creatine phosphate, snaps off that phosphate molecule, or excuse me, yeah, the phosphate atom, and it plasters it onto an ADP molecule, adenosine diphosphate, 2-phosphate. So when we add it to the ADP, now we convert that ADP into APP. So that's one of the ways that we can do it. But other than, because remember, keep in mind, glucose is the preferential fuel source for your cells. They love it. They love it. They love it. But it's not, how do I want to say it? You get a lot of ATP from glucose, but there are molecules that will give you more ATP or more bang for your buck. And the first one is listed here, fatty acids. That's why fat is such a good fuel source. Right? And that's why your body likes to store it, because it knows that it's a good fuel source that it can use, when needed on a rainy day. And so besides fatty acids and glucose, and we'll get into this in chapter 25 later on, lactic acid, amino acids, and ketone bodies, a lot of those will be shunted in through a couple processes, but ultimately gluconeogenesis. And remember, gluconeogenesis is going to be the creation of glucose from a non-carbohydrate source. Amino acids are a non-carbohydrate source. Lactic acid is a non-carbohydrate source. Right? Acetyl-CoA, all these things that you'll learn as we move through this class. So, all right, aerobic cellular respiration or aerobic metabolism is the best way, and that's pretty much what the heart depends on. But if you cannot supply the heart, right, with the oxygen that it demands, remember supply and demand, right, then we fall into a state of ischemia. And if it's long-term ischemia, you can eventually have tissue necrosis, where the tissue starts to get damaged, like in a myocardial infarction. Right? The tissues start to die when they don't have enough oxygen for metabolic needs there. And what can cause decreased oxygen? Interruption to blood flow. How about a blocked artery? Okay, pretty common. You've heard of people getting stents put in. Right? And so stents are just a way to keep those blood vessels patent, so blood can continue to flow through there. And I talked to you guys about the cabbage procedure. It's bypass, right? You're going to get rid of the garbage blood vessel, cut that out, right? Because there's probably some blockages in there. Put the new blood vessel in, graft it in, good to go. It's not that simple, but still. Okay, so that's the review that I wanted to talk to you about for myocardial tissue or heart tissue. So let's talk about the conduction system. So when you go to your doctor or nurse practitioner or whoever you go to see, and they put these pads on your chest and they check your heart rates with these pads, EKG, ECG, all right, that is monitoring the conduction system. Now it gives you a little bit more information than just what the conduction system do, but primarily it's going to be used to evaluate the conduction system of your heart. And so we're going to talk about that. So if we're talking about the conduction system, then we're going to be dealing with electrical events. Well, we talked about electrical events back in chapter 12, right? Remember that when we learned about action potentials? That's an electrical event. So that's essentially what we're going to be dealing with here. We're going to be dealing with nerve signals generated by action potentials. And so we have, all right, our cardiac muscle cells are going to be able to contract, but here's the thing. They can beat on their own. And we're going to talk about some of these. There's two different cells that we're going to be talking about. One, first, what I just mentioned was the cardiac muscle cells. They're going to be the contractile. And then we have this other cell called nodal cells. And these are the cells that are going to be part of this conduction system here. But here's the thing. The nodal cells do not contract. They don't. So they undergo this process called spontaneous depolarization. They're just going to keep depolarizing on their own. And we'll go through it and I'll explain it all to you. So when they depolarize, then they're going to trigger the cardiac muscle cells to depolarize. And then they'll contract. Nodal cells will not. Right? So essentially, you don't even need the nervous system, all right, to get contraction of the heart. But we need the nervous system, specifically the autonomic nervous system, to regulate the heart rate. Because otherwise, your heart is going to be beating so fast. So the autonomic nervous system, specifically, which division of the autonomic nervous system do you think slows the heart down? Parasympathetic. Yeah, the parasympathetic, the rest and digest. Parasympathetic will slow the heart down. Sympathetic will speed it up. Yep, good. So therefore, they have an antagonistic relationship when we're talking about the two divisions there, right? But we'll get into that. You guys will be pros by the time we're done with this chapter. Okay, so you see a bunch of terms up there. The first one at the top there is the sinoatrial node, known as the SA node. Its nickname, is it up there? Yeah, the pacemaker. So this is the pacemaker for your heart. So we're going to talk about the cells here that live in the SA node, right? But this kind of sets the rhythm. If you've ever been on a rowing team, I've never been, but there's a guy that sits on the boat that doesn't do any rowing, and he calls the cadence for the rows. That's the job that I want. I don't have to row. I just sit there and I tell everybody else to row. Fine. That's what the pacemaker does, all right? It sets the pace of your heart, all right? And so what's going to happen is pacemaker is going to depolarize. The action potential is spread out. It's like what I've told my classes before. It's like taking a stone and throwing it into a pond. And what happens, right? You get a splash, and then the ripples move out from the place where you threw the stone in. That's how your action potentials are in the heart. So the SA node will depolarize, and then the action potentials spread out, just run away from the center, and it goes across the atrial walls, right? So as it's spreading out, it's going to come to this structure here called the AV node, and this is on the floor of your right atrium. So that area between the right atrium and the right ventricle. So as it's spreading out, it hits the AV node, and then from there, right, you're going to have our nerve signals travel down, okay, into this structure called the bundle of His, also known as the atrial ventricular bundle. And if you notice, atrial ventricular is telling you the location. It's going to be between the atrium. I'll show you a picture here, between the atrium and the ventricles, and it's going to start to move down towards the interventricular septum there. And so that's what happens. It branches out, goes down through the interventricular septum. You get a left and right bundles as they head out, and then they'll head down to the apex of the heart, and then that's when we refer to those nerve fibers as Purkinje fibers, and they travel throughout the walls of the ventricles there. I'm going to come back to this slide. I'll show you. So here you can see, right? So there's your SA node, sinoatrial node. It depolarizes, and it spreads its action potentials in all directions, and they spread out like ripples on a pond, and then they come over here to the atrioventricular node, and then they cause the atrioventricular node to depolarize, and then it sends, now it's got its own little nerve fiber here, and so those are going to be the bundle of His here, the atrioventricular bundles, and then they'll split right when we get into about 1/3 of the way down in the interventricular septum here. Then they'll split into right and left bundles. And then when we get down to the apex here, then they just spread out all over the place. And you'll see them, right? They'll travel up. These nerve fibers will travel up through the ventricular walls, right? And those are known as Purkinje fibers. Because as we go through the class, we'll talk about bundle branch blocks and things like that. We'll talk a little pathology, but again, we're not going to be a lot of pathology in this class. So that's what we're looking at when we're looking at the conduction system here. So we're going to start up towards the top of the right atrium, and then in the floor of the right atrium, we have the AV node, and then that'll have those nerve fibers that will descend down into the ventricles here, and then just kind of spread out in all different directions there. Oops, wrong way. All right, questions, thoughts? I'll go over in this a little bit more detail. Yes. We'll talk about some of the rates. Yeah. Does anyone know what your range is for normal heart rate, heartbeat? 60 to 100 beats, roughly around 80. Okay. So 60 to 100. All right. Average is 80. Okay. So now that you've kind of seen the conduction system, let's discuss the nervous system, and specifically the autonomic nervous system that is going to help run this thing. So that means we have to go all the way up to your brain, specifically into your brainstem. But remember what I said, when in doubt, always go with the medulla oblongata, okay? So that's where we're going to go. We're going to go into the medulla oblongata. That's where the cardiac center is. And your cardiac center has two parts to it. Two parts. And it makes sense. Cardio-acceleratory, cardio-inhibitory centers. So take a guess. Which one speeds the heart up? Yeah, okay, good. So there, you don't have to memorize anything. You guys can figure that out. One less thing. All right, so we have our control center. That's going to be the cardiac center. Now, let's see the other parts of our homeostatic control system, the receptors. So we're going to talk about the types of receptors that we're going to see in the heart. And so you have two types, chemoreceptors. And if you recall, chemoreceptors are going to monitor, right, the composition of a solution. So in this case, the composition of the solution will be that of the blood. And then what are we going to see that we're looking for in the blood? Name me what levels of what are we going to monitor in our blood? That's a horrible question. What are we going to be looking at in our blood that is dependent on if we speed our heart up or down? If you're low or high in something. Not so much plasma. What's in our plasma? Well, water will play a role. Okay. All right, let's kind of go with that. Let me skip the chemoreceptors and go to baroreceptors. All right, baroreceptors monitor what? Stretch. If you drink a lot of water, where's that water eventually going to wind up? You said it. In the blood, okay? And what did I say? If you increase blood volume, what do you do to blood pressure? You increase the blood pressure, okay? And two things can affect the blood pressure, heart rate and total peripheral resistance. So what we'll see is when you get a stretch on the walls of the heart, okay, that is going to trigger your cardiac center, right, to pump blood out, right? So we can see an increase in heart rate. We'll talk about these reflexes later on, depending on what's going on. So if you get a change in the blood volume, which can change the blood pressure, that's going to be effectuated by the cardiac center. I don't want to get ahead of myself because I don't want to throw too much at you. Chemoreceptors monitor primarily oxygen levels in your blood, carbon dioxide levels in your blood, and then hydrogen ions. You remember what those are? Chapter 2, okay? Acids and bases sort of thing, okay? So we're going to talk about that. All right, so our cardiac center, when it's going to send out its motor output signals, remember, control center is going to interpret the data, and then it's going to decide what type of effect it's going to take. And so it's going to send that data or information to the effectors, which is a muscle or a gland, on our motor output, which are going to be the nerves that leave, all right? Remember the lower motor neurons and the upper motor neurons and all that stuff, right? So we're going to then send that information out through either the sympathetic or the parasympathetic division, depending on if we want to speed the heart up or slow it down. Right, so again, and I'll go through this. I'm just kind of hopefully trying to reawaken some of those memories that you had back in chapter 15, right, when we were talking about the autonomic nervous system there. Right, but notice, and I purposely use this word here, it modifies cardiac activity. It doesn't initiate it. Right, the cells in the heart initiate it. The nodal cells, when they depolarize spontaneously, the cardiac cells, they're going to initiate. The autonomic nervous system, specifically the parasympathetic and the sympathetic, are going to modify. They're either going to speed the heart up or slow it down. Right? And it can do two things. It can influence the heart rate and the force of contractions. So it can make your muscles in your heart contract harder. If they contract harder, they're going to be able to eject more blood out. And we'll talk about those. All right, this is in red for a reason, because everything on this screen is always important, but I really wanted to really have it stand out to you by putting it in red for a reason here. All right, so we're looking at our two divisions, and hopefully, and I think most of you probably remember, right, when you were in 210, you should have learned some of this stuff. What I mean is the parasympathetic nervous system slows the heart down, okay? So then it is going to be part of the cardio-inhibitory center. And if you recall, there are 4 cranial nerves that are part of your parasympathetic nervous system. Does anyone remember the 4? 3, 7, 9, and 10. 3 is ocular motor, 7 is facial, 9 is glossopharyngeal, and then one of my favorites, vagus, number 10. Okay, when in doubt, again, if you're not sure, vagus is always your answer. All right, if I ever ask you cranial nerve question for whatever reason, and you have no idea, go with vagus. Vagus does a lot. So vagus is part of the parasympathetic nervous system, and so the motor output signals that leave the medulla oblongata, your brainstem, are going to travel from your brain to your heart on the vagus nerve. You have a right vagus, you have a left vagus. All your cranial nerves are paired. So the right vagus goes to the SA node, and the left vagus goes to the AV node. Right? Cool, cool. Okay. The next part, sympathetic. Sympathetic, fight or flight. You know, when you have a near-death experience, like a collision or something, what usually happens? Your heart beats fast, right? For several reasons. Part of it is because you release some adrenaline, some epinephrine and norepinephrine, right? But your heart rate picks up. So sympathetic innervation, notice something here. It's going to increase two things. If you look here at the parasympathetic, it's only going to affect one thing. You need to know that difference. Probably going to ask you that on a test. Parasympathetic can only slow the heart rate down, whereas the sympathetic can increase the heart rate, but it can also make your heart contract harder, right? So it's going to affect your heart rate and then the force of contraction. Parasympathetic has nothing to do with the force of contraction, right? So sympathetic has two things that it can do. So obviously it's going to come from the cardio acceleratory center. And so if you recall, when you were learning about the autonomic nervous system and the parasympathetic outflow, which is the nerves that are leaving your central nervous system, I told you have the 4 cranial nerves, 3, 7, 9, and 10. And then you have the sacral division, which is S2, 3, and 4. Again, you learn that in 210. So the sacral division is 2, 3, and 4 for the parasympathetic. The sympathetic, you have the outflow that's going to be coming from T1 all the way to L2. So it comes through those, remember those spinal nerves that come off your spinal cord? Right, so you'll have some autonomic nerve fibers that go along those parasympathetic, go along those spinal nerves. So the specific ones are going to be T1 through T5. And so those nerves are going to travel to the heart, and you can see all of these places here, SA node, AV node, all throughout the myocardium and even the coronary arteries. Well, that makes sense, right? Because remember, only the sympathetic division can innervate blood vessels. So we need to change the diameter of our coronary arteries like any other artery, vasodilate or vasoconstrict. So the sympathetic nervous system is going to play a role in that. So T1 through 5 is going to have the sympathetic outflow, right, to those structures there. Here's an interesting fact. Those same segments innervate your chest and the upper part of your arm. Why is that? Good to know. Bingo. Yeah, Sam just nailed it. We call that what? You'll remember? Referred pain. Referred pain. And it's because the sensory neurons that innervate your skin in this area share the same elevator or ride share or whatever analogy you want to use as T1 through T5. They all hang out on the same nerve. Not the same exact nerve, but the same spinal nerves. So that's why, exactly, when you're having a heart attack, you'll have chest pain, left arm pain. Women will also get, for whatever reason, jaw pain along the angle of the jaw. All right. Questions about that? Does that kind of make sense to you? I hope it does. If I'm going too slow, let me know and I'll speed up. If I'm going too fast, let me know and slow me down. Good way to slow me down is asking me questions. All right, so know everything in red, but understand these concepts here. Okay, so this picture here is showing you, whoop, I don't know what I just did, y'all. I did what? There. Oh, okay. That was so nice of it. Okay, so, golly, I did it again. It looks so familiar here. Where is the thingy? Oh, there it is. Okay. All right. So I'll try to get this all in. Okay, so here you can see some of the outflow here. Here's the medulla oblongata with your cardio-acceleratory and cardio-inhibitory center here. And so you can see you'll have baroreceptors, which are the stretch receptors, and they're located in several different areas. You'll have baroreceptors in the heart walls. You'll have them in some of the blood vessel walls, and that's what we're seeing here. And then we'll also have chemoreceptors. And so the baroreceptors monitor stretch, chemoreceptors monitor the chemical makeup. And so they're going to send that sensory input information up to your cardiac center, right? Maybe your blood pressure is increased because you drank a lot of water, right? And so we've increased our blood volume, we increase our blood pressure. So we want to reverse that. And so there's ways that we'll do that, and we'll talk about it later in this chapter. But then the cardiac center will then respond to whatever those things are. And then if it needs to slow the heart down, then it's going to respond by sending that information through your vagus nerves. And the right one goes to the SA node. Slow the pacemaker down, okay? Inhibit it a little bit so it's not depolarizing as fast, so we can slow the heart rate down. If you slow the heart rate down, if your blood pressure is increased, you can decrease the blood pressure. Right? That's a perfect example of beta blockers. That's what they do. Beta blockers are a class of drugs that slow your heart rate down so you can decrease your blood pressure. And they also affect, right, I don't have it up there, our blood vessels, the lumen of the blood vessels. No, I don't have it there. All right, so we'll talk about this stuff. Just take a peek at this, right, just to kind of give you an idea how to map it out a little bit so you understand the anatomy a little bit. All right, so let's talk about how we stimulate our heart here when we talk about, all right, heart contraction. So we're going to start off here by talking about the conduction system here, and then we'll talk about the cardiac muscle cells there. So we're going to start off with the nodal cells, and then we'll dip into the cardiac muscle cells. So keep in mind, remember I said the autonomic nervous system modifies the heart rate. Right? The conduction system is what initiates it. All right, when those cells, which we'll talk about here momentarily, when they start to depolarize, they're going to be initiating this process there. Right, so the conduction system is going to initiate it, and when I say it, we're talking about the action potentials. And then the cardiac muscle cells, they too are going to create action potentials. But here's the thing, these cells will contract. So we're going to see depolarization first in the atria and followed by the ventricles. So when we learned about muscles, what usually happened after we caused the muscle to depolarize? Well, when we depolarize a muscle, right? So you saw the neurotransmitter crossed over the synaptic cleft there, and then it depolarized the end plate. We called that the EPP, end plate potential. So we got a little mini action potential. And if it was large enough to exceed the threshold, which was, do you remember? Well, in nerves, it's 70. All right, remember sorry, in nerves, it's 55. In muscles, it's 10 points higher, so 65, negative 65. Okay, so when we got to that threshold of negative 65 millivolts, then we got a full-on action potential. Okay, that's great. An action potential is great, but it's not a contraction. So the action potential occurs, and we know the whole series of events. We release calcium from the sarcoplasmic reticulum, and then it goes into the sarcoplasm. It removes troponin and tropomyosin off of the binding site for actin, all right, for myosin on actin, and then we can get our cross bridging. So what follows an action potential? What follows depolarization? When we're talking about muscles, a contraction, okay? So normally you're going to get depolarization followed by repolarization, right? And that's what we're going to see here. Same thing, doesn't change, but after you depolarize a cell, it's going to contract, right? That's what we're looking for. So that's what we're going to see here. We're going to cause the cardiac muscle cells to contract. So depolarization initially starts in the atria, so they should contract first. And then the ventricles will depolarize after the atria, so they'll contract after, which is good. Because remember, atria, when they contract, what are they doing with their blood? They're pushing it down into the ventricles. And then when the ventricles contract, what are they doing with their blood? Pushing it into the arteries there, into the pulmonary trunk and into the aorta. Good. All right, so here's that nodal cell I was telling you about. So you have all these nodal cells sitting in the SA node. Keep in mind, they initiate the action potential there. Like I said with that analogy, it's like throwing a big rock into a pond and then the ripples just go out from there. And that's what those action potentials do. You can see here, right, with all these arrows, the action potentials will spread from the right atrium to the left atrium, but also they'll go inferiorly down towards the AV node there. And so as we depolarize, we're going to see, all right, contraction following that. Right, so these are the guys, these cells here are going to initiate that process. Good so far? Okay. Then with our cardiac muscle cells here, right, they're going to have their action potential, which is going to cause them to depolarize. And when they depolarize, they're going to be followed by a muscle contraction. So that's what we'll see, right? Those cross bridges forming, and then we'll see that shortening of the sarcolemma, not sarcolemma, excuse me, sarcomere. Right, so again, we know this process. I'm just kind of reviewing that because that's what we're going to see. Nodal cells do not contract, right? Our cardiac muscle cells do contract. Both cells depolarize. This cell depolarizes with an action potential, and this cell will depolarize, except one contracts and the other one doesn't. Okay. So let's start off like we did in chapter 12 when you were learning about neurons. We're going to start off with everything at rest. You're not doing anything. Everything's just hanging out and living life and being fine. So what we're going to see is the SA nodal cells are going to be doing this process. Spontaneously depolarizing. What does spontaneously mean? Yeah, it doesn't need any help. It just does it. Okay? It just keeps depolarizing. That's what it does. All right? So that's what it's going to do. So that means if it's going to constantly depolarize, what follows depolarization here? Well, I know I said muscle contraction, but an action potential gets generated. That's the whole point of depolarization. We're going to create a nerve signal. And that's what these cells do. And so another number for you to know, okay, we learned that in our neurons, our resting membrane potential is -70. So here, when we're talking about our nodal cells, it's -60. So it's a little bit less. Okay, so it should be a little bit easier, depending on what's happening, but it's a little bit easier to get depolarization there. All right, so know this value because it makes it a little bit easier when I'm trying to use numbers here to explain stuff for you. All right, so this term here, pacemaker potential, ability to reach threshold without stimulation. Think about that. Remember what I was saying, that these cells, what are they doing? They're spontaneously depolarizing. And we agree that that means they don't need any help. All right, so that's what that pacemaker potential is going to do. We're going to be able to reach that threshold, right, which is going to then cause depolarization, create an action potential, and then that action potential will stimulate our cardiac muscle cells to depolarize, and then they can contract. All right, that's what we're trying to do. So when we are looking at these cells, a lot of this stuff you already know. Right, we're going to be looking at the plasma membrane because that's where those action potentials occur. So there's some transport proteins. And we saw in the neurons, we had sodium gates, we had potassium gates, we had leak channels and all sorts of fun things, right? You're going to see the same thing here. So we got our sodium potassium ion pumps, and we know what they do, right? Sodium potassium ion pumps, they pump sodium out of the cell, potassium into the cell. 1 ATP gets you 3 sodium kicked out of the cell, and it gets 2 potassium brought into the cell, just with 1 ATP. We have our calcium pumps. We saw that before, okay, down by the transmissive segment, by the synaptic knobs. And those calcium pumps, their job was to pump calcium out of the cell, right? And so by doing that, it creates a membrane potential. And then finally, we have our leak channels. The leak channels, those are the proteins that are most responsible for a resting membrane potential in a cell. Hopefully you remember that. Right. In a neuron, the reason why it has that -70 resting membrane potential primarily is because of the potassium leak channels. The sodium leak channels contribute to that a little bit. And then our sodium potassium ion pumps help to get it to that -70. But primarily the leak channels are going to be responsible for that resting membrane potential there. So something new now that we're going to see in our nodal cells is something called a slow voltage gated sodium channel. We saw voltage-gated sodium channels before, but never slow. And then we also have fast voltage-gated calcium channels. That's important. So we give the descriptors fast and slow because when we look on this graph that I'm going to show you shortly, it'll make sense as to why it looks the way it looks. Right? So make sure that you learn and know these slow voltage-gated sodium channels, fast voltage-gated calcium channels. And then, of course, we got our voltage-gated potassium channels. We had those before. Right? So just kind of two new channels that you didn't know before. So when you're looking here, now mind you, keep in mind, we're still at rest. Heart's not contracting yet. Nothing's going on. Right? So this is what it looks like here in the plasma membrane for these channels here. You got your voltage-gated cation channel, and then you got your voltage-gated calcium channel, and then you got your voltage-gated potassium channel. Right? So this is what we're seeing here in the plasma membrane all around the cell. So keep in mind, what did I say in the previous slide here? Calcium pumps, they're going to pump calcium out of the cell. So that means you're going to have more calcium out of the cell than inside the cell. That's the purpose of pumps. They pump, all right, whatever they're named after, they're going to move things against their concentration gradient. So if you recall, your sodium potassium ion pumps, yes, they pumped sodium out of the cell, but they had to pump it because there's way more sodium outside the cell. You heard me mention banana floating in the ocean before, okay? So banana, it's going to be potassium, that's the cell, and then sodium and chloride are outside the cell. So here's our nodal cell, same kind of scenario. You got lots of potassium inside the cell, sodium's going to be outside the cell, chloride's going to be outside the cell, and now calcium's going to be outside the cell. We want more of that outside and more of this on the inside, so we create that concentration gradient. So when we start opening up these channels, you all know diffusion, right? Where you're going to have a solute or a substance move from an area of high concentration to low concentration. And that's what these channels do. They act in what we call facilitated transport. They're going to help with that process there. So let's start off. I'm going to tell you a story. I even have it in here in 3 parts. Okay. So the first thing you see way at the top here, SA node cells exhibit autorhythmicity. Spontaneous firing. They're just going to keep depolarizing. Right? And they're going to go through these processes here. Right? So we got our threshold, depolarization followed by repolarization, then rinse and repeat. That's what we're going to do. So when we talk about reaching our threshold, here's how it happens. We're going to start off with our slow voltage-gated sodium channels, like we saw in the neurons. So those will open up, okay? But notice the term slow. That means that sodium is slowly going to enter the cell. So what does that do to the inside of the cell? When sodium enters in, what does it do to the polarity of the inside of the cell? It makes it less negative, right? And what's that called? Depolarization. Yep, depolarization. When you make the inside of the cell less negative, or you could say more positive, that we're starting to depolarize the cell. So these open up, sodium flows in. Now we go from -60 to -40. Boom, we hit our threshold value. As soon as that happens, now that's the stimulus that causes the fast voltage gated calcium channels to open up. And when that happens, where's more calcium? Outside the cell or inside the cell? So where's calcium go? Inside the cell. Exactly right. Calcium flows in and it flies in. So it takes it the rest of the way. It goes from that -40 to 0. The inside the cell now, the inside the cell is less negative, more positive. We have now done depolarization. So once we get to this value here, that stimulus causes your calcium channels to close, triggers the opening of the voltage-gated potassium channels, and you guys know the story. Now, that positive ion, potassium, that cation inside the cell, will leave the inside of the cell, take its positive charge with it, thus making the inside of the cell more negative. That's repolarization. So we go from 0, and as potassium flows out, taking its positive charge with it, we go from 0 to -10, -20, and so until we get back to our -60. And then we repeat the process. We get to -60, bam. It causes the slow voltage sodium channels to open up. I think I got a picture of it. I'll show you here, right here. So let me show you with pictures. All right, so step 1, reaching threshold. We're at -60. Okay, that's our resting membrane potential. Right. This value, that's our stimulus, triggers the slow voltage gated sodium channel to open up. Lots of sodium outside the cell, not a lot inside the cell, causes sodium to enter into the cell, bringing its positive charge with it, therefore making the inside of the cell less negative, right? Does that make sense? Okay, so that's what we see here. So we go from -60 to -40. There's our threshold value, right? Now that's going to trigger, now that's going to cause the fast voltage gated calcium channels to open up. Calcium then pours in, and it gets us to that 0 millivolt value there, okay? And then when that occurs, we get to this value here, then this gate closes down and it triggers the opening of our voltage gated potassium channel. Potassium leaves the cell, takes its positive charge with it, and that then starts to make the inside of the cell more negative. That's our repolarization. Once you get to this value here of -60, we repeat the process. The potassium channels close. We go all the way back over here to our slow voltage gated sodium channels. They open up, same process. So when we graph that, right, here's what you're seeing. So if you see this when you're studying, now you can understand when we're tracking, right, the polarity here and the charge. You can see at resting membrane potential. Once you get to that -60, that triggers those slow voltage gated sodium channels to open up, and it, I won't say trickles in, but it doesn't move in very fast. That's why you can see this line is a little bit more diagonal. Once we get to our threshold value of -40, then that kicks into gear the fast calcium channels to open up. And that's why when they flow in, you can see a huge vertical spike there. Happens almost instantaneously. We get around that 0 charge there, causes this channel to close, causes the potassium channels to open, and then potassium leaves the cell, and then we start to repolarize the cell. Once we get to -60 again, repeats itself. Nothing's telling it to do this. It just does it. Yes, ma'am. It has to do with the concentration gradient. Remember, concentration gradient, when we talk about diffusion, there's 2 things that can affect that. Temperature, which we're not even talking about, and how steep that concentration gradient is. Like how much is coming in, okay? So again, plus you're going to still have inside the cell, right, some calcium in there. It's not going to be devoid. So you'll see, well, I shouldn't say, but you have a lot more outside the cell because you've got those, so the calcium pumps are pumping it out. So it just has to do with that concentration gradient, the difference there. All right, so I didn't talk about this slide here. So I think, Christian, you made mention about the heart rate and whatnot. So this slide is important, all right, because like I said, our normal heart rate is between 60 to 100 beats per minute, usually around 80. Varies from individual to individual. So here's what happens when the SA node depolarizes on its own, right? Its action potential is slightly less than a second. And that should be about 75 beats or 80 beats a minute. But here's the thing. You don't see that. When it's just the SA node that's influencing your heart rate, the SA node depolarizes way fast. And so you get about 100, right? So that's what you would actually, what you're going to see is if it was up to the SA node, your heart rate would be a lot higher than what it presently is when you're at rest, when you're not even doing anything. 100 to 110, 120 beats, because it depolarizes that fast. Boom, 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 boom. Right? But what allows us to maintain our normal heart rate is this guy here. And we talked about this back in 210, right? Vagal tone. It has to do with the vagus nerve that is going to slow things down. And that's what the vagus nerve does. Remember, rest and digest, parasympathetic is going to be antagonistic of the sympathetic nervous system. Sympathetic speeds up, parasympathetic slows down through the cardioinhibitory center. And that's what happens here. This is essential. So when during a transplant operation, when they disconnect the actual heart, the nerve from the heart, it will start to beat fast, much faster than what it normally would beat there. And so this here, the vagal tone, is very important, so we're not burning through energy. So keep in mind, when we talk about this autorhythmicity, that spontaneous firing, that returns us back to that pacemaker potential here, right? The ability for us to reach the threshold with no stimulation. We don't need the nervous system to affect those nodal cells. Right? What causes that is just depending on what the concentration amount of sodium in and outside the cell and the concentration amount of calcium inside and outside the cell. So keep that in mind. When we talk about pacemaker potential, we don't need any help to get to that threshold value of what? Do you remember? It's not up there. Threshold. Well, negative 40, right? And then your resting membrane potential for a nodal cell is going to be negative 60 there. Okay, so let's return back to the conduction system. Back to the conduction system. SA node, AV node, bundle his, and all that stuff. So I'm going to kind of trace this out for you, right? Let's just put it into some words here first. All right, so our SA node will spontaneously depolarize, and so we generate that action potential. So that's literally throwing, not literally, but it's like throwing that rock into that pond. So we throw the rock into the pond, we generate our action potential, and so those action potentials spread out throughout the atria first. And then it's going to eventually make it to the AV node here. Keep in mind, this is where those those intercalated discs come into play. Right? Remember, we talked about the gap junctions. They're going to allow the movement of those ions that I just got done talking to you about. Right? So meanwhile, you have to understand, I didn't really talk about this. You got ions flowing from the outside of the cell and into the cell, and vice versa. You got ions from the inside of the cell flowing out. But I don't really talk about it. But at the same time, you got ions flowing from one cell to the next cell to the next cell because of those gap junctions. So we can get ions to help depolarize the cell out from outside the cell, and then we can get those ions that can flow from one cell to the next neighboring cell. So we'll see that there. And that's, again, this whole concept, those gap junctions allow for that synergy, that ability for those two atria to contract at the same time. I can't stress to you enough how important it is to make sure that it contracts at the same time and it doesn't vary. Because if you have alternate contractions, or if it's off a little bit, you'll get a regular heartbeat, and you can start to get damage to tissues and whatnot. Okay. So SA node discharges, depolarizes, it sends action potentials everywhere, and then those action potentials arrive here at the AV node. This is very important that you understand this. We want to slow things down at the AV node, right? By design. Kind of a cool thing to happen, because we still need the ventricles a little bit more time to fill up, and then they have to start their contraction process. So you can't have the atria done contracting and then right afterwards the ventricles. It'll look like it when you watch it, but for a brief moment there's a little bit of a delay. Remember that isovolumetric contraction when all the valves are closed, right? So we need to just have a small brief delay. And so that's what the AV node does. It literally will act like a bottleneck, because the way it's designed is there's very few gap junctions in and around the AV node. So we're almost forcing all that electrical current. It has to go to the AV node before it travels onto our bundle branches and whatnot. So we want to keep what causes it to slow down. Y'all remember how we can increase action potential speed in our neurons? Make them bigger, and what's the other one? Myelination. Yeah, that's right. That's right. That's how you speed up action potentials, right? You myelinate the nerve, or you have a bigger nerve, like a pipe. You want to get more water to flow through a pipe, you have a bigger pipe, all right? So specifically in this area, we have a very narrow or small diameter neurons there, so that slows it down. And guess what? We're going to reduce our gap junctions so ions can't be shared as well between neighboring cells. Bottleneck gives our atria time to finish contracting before the ventricles start to contract. That's very important here, right? And so once we move from the AV node here, now we're going to move our action potentials from the AV node down to the Purkinje fibers. So they'll go from the AV node to the AV bundle, and then that bundle will split into left and right bundle branches. And then we get down to the apex of the heart, and that's where we form our Purkinje fibers. And the Purkinje fibers will then come from the apex of the heart and spread upwards, right, like vines all throughout the heart walls of the ventricles there. Right? And so we'll see tons of gap junctions in this area here, because gap junctions will help to increase that conduction for the action potentials, and the neurons will be a little bit larger, so we'll see an increase in that, in the conductions there. Because keep in mind, our goal with anything is to cause simultaneous contraction between the two chambers. Ventricles need to contract at the same time, and the atria need to contract at the same time. We don't want the ventricles and the atria contracting at the same time. And so that's what that AV node does. It helps to prevent that. So let me show you here in picture form. Okay, so here you can see the beginnings of our action potential. Here's our SA node. Nodal cells spontaneously depolarize, right? Sending action potentials throughout both of the atria, right? Those action potentials will make their way down here to the AV node, right? At the AV node, they get slowed down briefly, fewer gap junctions, smaller neurons there, so it slows things down there, right? That allows the atria to contract before the ventricles start to contract. Now, our action potential makes it from the AV node to the AV bundle, and it starts to descend into the interventricular septum here, and then it'll split into left and right bundle branches there. So our action potential starts to move down here. Into the bundle branches. And so those start to get a little bit bigger. We're starting to see more gap junctions. So the bundle branches split, the bundle splits into the left and right bundle branches. We get down here to the apex. That's where the Purkinje fibers are. And then from there, because our atria have now finished contracting, this all happens during that isovolumetric contraction, or just prior to that. Then we start to see, all right, our Purkinje fibers are going to start to depolarize your cardiac cells from the bottom up. So we see depolarization starting here at the apex and working its way up. And if you remember in the video, the apex of the ventricles starts to contract first, which makes sense, bottom-up contraction to push the blood up and out of the ventricles into our great vessels here. All right, thoughts, questions, concerns on anything? Okay. All right, a little bit more on the Purkinje fibers. Like I said, they are going to be larger in diameter. And as we know, larger diameter blood vessels, larger diameter nerves or neurons are going to move an action potential much faster. And this helps to keep our ventricles, like the trains, running on time and contracting at the same time here. So some of these Purkinje fibers roll into the papillary muscles here. What's the purpose of the papillary muscles? Not up there. Well, y'all remember? Well, what do the papillary muscles attach onto? Yeah, exactly. Yeah. So, because the papillary muscles are going to contract to keep the valves from opening up. Because at this point, the AV valves are closed. And when those ventricles start to contract and we increase the pressure inside of those chambers, we don't want them to blow the AV valves open in the wrong direction. So the papillary muscles are going to start to contract. They're going to yank on the chordae tendineae, which are attached onto the atrioventricular leaflets or cusps of the valves there. And it helps to make sure that blood does not release. So there will be a contraction there. And like this says here, our contraction at the ventricles starts at the apex there. And so apical contraction first, and then it moves superiorly up through the ventricles, pushing the blood upwards into the arterial trunks there. First clinical view of the day, ectopic pacemaker. Has anyone ever heard of that? No. So something else is going to be the pacemaker of your heart. Okay, the SA node, in this case, will not be. So the pacemaker will be something other than the SA node. All right, so the SA node is nice because it has the ability to spontaneously depolarize. Guess what? We can also, if we take out the SA node, right, now we're going to see something, because the SA node, remember I told you, it wants to beat over 100 beats per minute. If it's left alone, that's what it will do. So if we take out the SA node, we have a new pacemaker involved, and we default to the AV node. Thing with the AV node is it's a little bit more laid back. So it's going to have a much slower spontaneous depolarization rate, and that's going to be anywhere between 40 to 50 beats per minute. You can still live on that. In fact, there was a cyclist, a professional cyclist. Anyone ever heard of Miguel Indurain? This is a long time ago. It's like 30 years ago when I was in college. This guy had a resting heart rate of 27 beats per minute. Insane. 27 beats per minute. Okay, that was his resting heart rate. Right. So obviously, yeah, and he lived. Yeah. So that's a lot less than 40 to 50 beats a minute. So you can live on that. But now if we take out the AV node, now we're left with just the cardiac muscles. Remember, I told you they can depolarize on their own, but they're way too slow. I mean, unless you're a highly specially trained athlete, which no one in this room is, as far as I know. No offense to anybody. Sorry, Sam. All right. No, I'm kidding. I mean, that's extreme. I'm talking about you're in shape. Okay. This is not going to be enough. And so you're not going to make it. But so you have backup systems. If the SA node is no longer able to be the pacemaker, you default to the AV node. If the AV node can't keep up with it, that's when we have to. What's the cure to this? Yeah, electricity. You're going to put a pacemaker in. You're going to put an actual pacemaker into the person's heart that will help to reestablish the proper rate there. All right, I won't be able to finish this today, but I do want to talk about cardiac muscle cells. So that's just the nodal cells. That's just the cells. All right. Again, kind of review that. Listen to the video a little bit. If you need a little bit of help on that. Cardiac muscle cells, you're going to be a little bit more familiar with because you've already seen them. You already know. You learned about them in chapter 10. All right. So let's start off talking about the anatomy of these structures here. So same players here. Sodium potassium ion pumps. That's going to help with our concentration gradient. We're going to have the calcium pumps, and then of course our leak channels. All of these are the same. Right? We saw those in neurons, we saw them in nodal cells, and now we're going to see them in cardiac cells. And so their job is to create that concentration gradient. And if you don't know that, make sure that you know that. Right? These cells, that's these transport proteins, that's their job. It's to set up this phenomena over here on the board. Banana floating in the ocean with some calcium outside. Right? That's what we're trying to do. So here, we have our resting membrane potential of -90. So we've seen this before. We start with a resting membrane potential, and then we have to go from our resting membrane potential to our threshold value. That's what we're shooting for, okay? So how is that going to happen? We're going to use these other transport proteins, these voltage-gated channels. In this case, we have two new ones. We saw in the nodal cell, slow voltage-gated sodium channels and fast voltage-gated calcium channels. Well, in the cardiac muscle cell, we're just going to flip them. So now you're going to have fast voltage-gated sodium channels, slow voltage-gated calcium channels, then of course, potassium channels, same old, same old. So in cardiac cells and nodal cells, we're just switching the fast and slow with the channels. Just don't get them confused, right? Learn one, then know that the other cell is opposite. Right? So when we're dealing with a cardiac muscle cell, like we saw with a neuron, right? all of the gated channels are closed in the neuron at rest. Same thing here. Our voltage gated channels are all going to be closed at rest. So here's a picture of those gated channels at rest. So here's our cardiac muscle cell, just chilling out, minding its own business, -90 resting membrane potential. All of these gated channels are closed, but guess what's still open? These guys. When I say open, they're still working. The leak channels are always open, right? Like 7-Eleven, open 24/7. Leak channels are always open, right? And then our sodium potassium ion pumps and the calcium ion pumps are pumping. So when all this other stuff goes on that we're going to be talking about, when a cell depolarizes and when a cell repolarizes, these proteins are doing what they always do. It does not change. I want you to understand this is going on in the background. So when we start to open up these gates here, these channels and pumps are still doing what they do. Okay? Keep that in mind. That's all. This never changes all the time. We don't talk about it because we just assume y'all know that's what it's doing. All right? So keep that in mind. All right. So let's talk about depolarization of our cardiac muscle cell. So same type of scenario. Now we've added a few things here that are going to be a little bit different. But first, we got depolarization followed by this phenomenon called plateau, and then it's going to be followed by repolarization. So something new, except we just inserted something in between, all right, depolarization and repolarization, which I'll talk about here. All right, so let's start off with depolarization. Same thing. We're going to be, our resting membrane potential is going to start off at that -90. So in this situation, right, we get, right, from the conduction system, or remember those gap junctions there that are allowing ions to be shared, right, between cells. But point being is we start to see, right, the triggering of reducing the threshold, the resting membrane value. We're going to start to approach that threshold value. So we're going to see, right, the impulse here, which is going to trigger this fast voltage. When I say fast, it's going to look like this on our graph. Here's our graph. Look at that. So when our fast voltage gated sodium channels open up, that's pretty much vertical line right there. That's how fast that this cell goes from -90 to a +30. All because this gate, not that one gate, but many gates like it, once that opens up, so much sodium flows into the cell that it literally causes a pretty much vertical line of depolarization. And that's just from sodium going into the cardiac cells. Right, so sodium enters in, we go from -90 to +30. Right, and so that +30 then is going to then stimulate this phenomenon called the plateau phase. See on our graph here, it's kind of almost horizontal, sort of. Right, so that's our plateau phase. So once we get to that +30, then the slow voltage gated calcium channels open up. And in this case too, now we're going to also open up our voltage gated potassium channels. So this is the first time that we've actually seen the voltage gated potassium channels start to open up prior to repolarization. So you have two positive cation channels opened up. But here's the problem. Potassium's on the inside of the cell, calcium's on the outside of the cell. So these two gates are going to allow ions to flow in opposite directions. So you're going to be gaining calcium from outside the cell. You're going to be gaining positive cation ions from outside the cell, but at the same time, you're going to be losing positive cation ions from inside the cell. That's why the polarity of the cell does not change all that much. I mean, it's still repolarizing. It's still becoming more positive on the inside, and that's only because you're getting more calcium coming in than potassium leaving, right? But we're still getting that kind of plateaued horizontal stage there. So keep that in mind, right? So potassium leaves, calcium enters there, and then don't forget, we're getting a lot of that calcium, not just from outside the cell, because the cells have been pumping it out through the calcium ion pumps. Don't forget, I haven't talked about it yet. Well, I talked about it earlier, our sarcoplasmic reticulum. That's where we get most of our calcium. It's stored there. And so the action potentials are opening up those calcium channels on the sarcoplasmic reticulum that are going into our cardiac cells. And not only that, the cardiac cells themselves have their own voltage-gated calcium channels. So we're seeing a lot of that calcium going into the cell, more coming in than potassium leaving. But at this point, we are still depolarized because the inside of the cell, right, is still below that threshold value. It's still more positive. All right, and then after all that happens, then the voltage-gated calcium channels close, but the potassium channels stay open until we get to that -90. So it's going to look like this. So once those calcium ion channels close, then potassium keeps pouring out of the cell. Nothing more positive is coming in, so we're able to repolarize, and so our line goes more vertical, going back down. So let's go through step by step here. All right, depolarization, just to review. Well, actually, let's review this. All the gates are closed right at rest, right? And our resting membrane potential is -90. Okay, so now we get that electrical signal that depolarization is occurring. Our action potential comes in. And so that action potential then triggers the opening of the fast voltage gated channel. Sodium pours in, and it causes that, right, that huge spike there. So sodium pours in. Once we get to this value of +30 or in and about, that's the stimulus that then causes our potassium voltage gated channel to open up and our slow voltage gated calcium channels to open up. At this point, the fast sodium gated channels have closed, potassium leaves, calcium enters, but more calcium is entering the cell than potassium is leaving. Right, so we're going to see a downward, a decrease in the positive charge inside the cell. We're starting to make the inside the cell more negative, right, as this is occurring. And that's our plateau phase. That's this part. That one there. And then what will happen, the last part for repolarization, then our slow gated calcium channel closes down, and the only thing that's left open is the potassium channel. The potassium still leaves until we get to that -90, and then that's when the potassium voltage gated channel closes down. So be familiar with this. And in fact, I got a slide on here. Oh, shoot, wait, hold on. I thought I had it on. I don't have it on here. I thought I did. But I want you to be able to tell the difference between, right, what the graph looks like for the depolarization, repolarization of a cardiac cell versus a nodal cell. Okay, you want to know why it looks. These pictures here will tell you why, because they're numbered to show you what's happening at each of these phases. And this slide here, what figure is that? That is figure 1919. This one here, same thing. It tells you what's happening at each of these stages there. All right, so just be familiar with that, okay? Because this will help you, because when we talk about certain drugs like calcium channel blockers, I'm sure some of you may have heard of that. Well, guess what channel we're blocking, right? Those channels. That helps with that depolarization, because what follows depolarization? Not other than repolarization? Yeah, contraction, muscle cell contraction. All right. I'm going to stop here. Yep, let's stop here. And you got a question? All right, hit me. What do you mean, like a... Right. Yeah, they'll stay open for a short period of time. Yeah, and then they'll start to close down. Yep.